So I am in your living environment with Ms. Trantham, and I'm looking at the experimental design assignment. I'm going to read through the big idea, objectives, facts, and examples. That's the green text. And then on a piece of paper, you're going to write your responses to the blue text and then take a picture of it and send it to me, or you can send it to Ms. Trantham. It starts with big ideas. The central purpose of scientific inquiry is to develop explanations of natural phenomenon in a continuing and creative process. Beyond the use of reasoning and consensus, scientific inquiry involves the testing of proposed explanations involving the use of conventional techniques and procedures, and usually requiring considerable ingenuity. The observations made while testing proposed explanations, when analyzed using conventional and invented methods, provide new insights into natural phenomena. Objectives: devise ways of making observations to test proposed explanations. Develop and present proposals including formal hypotheses to test explanations. Predict what should be observed under specific conditions if the explanation is true. Based on the results of the test and through public discussion, revise the explanation and contemplate additional research. Important facts. Fact number 1. Scientific inquiry. You observe. You come up with a question. Form a hypothesis, which is a prediction based on research and observation. The hypothesis must be testable and written in a statement form. If blah blah blah, then blah blah blah. 4. Conduct a controlled experiment. See Fact 2. That will be down here. Record and analyze data. See Fact 3. 6. Draw a valid conclusion from data. You are either going to accept your hypothesis, modify your hypothesis, or reject your hypothesis. Number 2. Controlled experiments. Grouping. You will have an experimental group, the group that is being tested, getting the actual experiment. A controlled group, that is the group that is not being tested. It's unchanged, but rather is being used to check the validity of the experiment, may be getting a placebo. Remember, the placebo effect is where one group got a sugar pill, the other one got an active medication, but because of the fact that they think that they're getting the real pill, they're showing symptoms, and they're all of a sudden better, when in reality they're not. Their brain's tricking them. 3. Variables. Controlled variables. The variables that are kept the same between the different groups so they don't affect the experiment. Independent variable. The variable that the researcher is testing or changing on purpose. When graphed, it goes on the x-axis. Your dependent variable is the variable that changes as a result of the independent variable, what you're changing on purpose. When it is graphed, it goes on the y-axis. Ways to increase validity: increase your sample size and repeat experiments with same results. You can also have your experiment be peer-reviewed. Have other people test it. Example: if I have a question that says, what is the effect of fertilizer on plant growth? My hypothesis says, if plants that are given more fertilizer will be taller than plants given no fertilizer. So my control here is the fertilizer. My experimental group, 25 green bean plants, will be given 2 tablespoons of fertilizer. 25 green bean plants will be given 4 tablespoons of fertilizer, and 25 green bean plants will be given 6 tablespoons of fertilizer. Control group is the 25 green bean plants that will be given all the same conditions except they will get no fertilizer. So there's actually 4 groups. The first group has no fertilizer. Second group has 2 tablespoons of fertilizer. Third group has 4 tablespoons of fertilizer. And the fourth and final group has 6 tablespoons of fertilizer. Controlled variables. Amount of soil isn't going to change for any of the groups. The amount of water given isn't going to change for any of the groups. Amount of sunlight isn't going to change. Temperature isn't going to change. And the amount of time for the experiment. They're all going to get the same amount of time to grow. The thing that will be changing, the independent variable, is the amount of fertilizer each of the groups get. The dependent variable is the plant height. It's going to change, hopefully, based off of our hypothesis, with the amount of fertilizer that we use. The data collected is the height of the plant will be measured 1 time each day for 10 days. Data recording. Data will be recorded in a data table and plotted on a graph. Conclusion. If my hypothesis is supported by the data, the plants receiving 6 tablespoons of fertilizer will be the tallest in our groups. Okay? So based on the green information, you are going to be responding to the blue questions. If you have a computer, you can go and copy and paste this and respond to the questions by just typing underneath it. If you don't have a computer, write down the question number, write down your answer, and you can take a picture of it and send it to Ms. Trantham. Base your answers to questions 1 through 5 on the information below and your knowledge of biology. Poison ivy is a weed that grows in New York State. It synthesizes an oil, urushiol, that causes skin rashes. Researchers have found that if poison ivy grows in an environment that contains an increased concentration of carbon dioxide, the plants grow larger, faster, and produce more urushiol. Because carbon dioxide levels in the atmosphere are rising, poison ivy might become a hazard to people who work or vacation outdoors. Question number 1. State the hypothesis the experiment would test. What are they assuming is going to happen? Number 2. State one way the control group should be treated differently from the experimental group. Number 3. Identify 2 conditions that should be kept the same in both the control and the experimental groups. Describe the type of data to be collected. Number 5. Identify one safety precaution that should be taken during the experiment and explain why it is necessary. Number 6. An experiment was carried out to determine how competition for living space affects plant height. Different numbers of plants were grown in 3 pots, A, B, and C. All 3 pots were the same size. The data collected are shown in the table below. Our title says Average Daily Plant Height, and it goes up to day 7. Pot A has 5 plants. Pot B has 10 plants. Pot C has 20 plants. Analyze the experiment that produced the data shown in the table. In your answer, be sure to state a hypothesis for the experiment, identify one factor other than pot size, because we know that was kept the same, that should have been kept the same in each experimental group, identify the dependent variable, what is being changed based off of our independent factor, state whether the data supports or fails to support your hypothesis, and justify your answer. Base your answers to questions 7 through 9 on the information below and your knowledge of biology. A study of antibacterial cleansers. An experiment was designed to test the effectiveness of 3 antibacterial hand cleansing solutions against bacteria present on hands. Swabs were used to take 1 sample each from the unwashed hands of 10 test subjects. Each swab was then rubbed across the surface of bacterial growth medium in a separate petri dish. The dishes were placed in an incubator to allow bacterial colonies to develop. 10 other test subjects treated their hands with an antibacterial hand cleansing solution, then had their hands swabbed, and 10 more petri dishes were set up and incubated in the same way as the first set of dishes. The process was repeated again with 10 more subjects for a second hand cleanser, and again for a third group with a third hand cleanser. The results of the incubated petri dishes were averaged, and the averages are shown in the table below. The table is titled The Effectiveness of Antibacterial Cleansers. It's got a treatment before swabbing and the number of bacterial colonies recorded. Number 7. What was the purpose of testing unwashed hands? Number 8. Explain why the researchers used data for 10 trials and averaged them rather than data from a single trial. Number 9. Based on the data provided, what could the researchers conclude regarding the effectiveness of the antibacterial hand cleansing solutions tested? Base your answers to questions 10 through 12 on the information below and your knowledge of biology. Ticks, such as deer ticks and dog ticks, feed on the blood of humans and other animals. Part of the feeding process involves the tick injecting its saliva to help make blood flow. In the process, they sometimes spread disease organisms to their hosts. Sometimes ticks get on clothing and can remain there for a few days before actually biting their host. A scientist found that ticks might be able to survive even when exposed to hot water and detergent in a washing machine. Students designed the experiment below to test how well ticks survive a hot water washing machine cycle with detergent. Note that some details of the design are incorrect. Hypothesis: can ticks survive hot water and detergent washcloth cycle in a washing machine? Data can be collected: number of ticks that survive the cold water wash cycle. We got our control group, deer ticks, cold water and detergent wash, and the number of ticks is 10. Experimental group, dog ticks, hot water and detergent wash, and 100 ticks are tested. Identify one error with the hypothesis. Explain why students proposed data to be collected would not likely test what the students intended. 12. Identify one error in the student's design in the shaded area of the table and explain how the students should change the experiment to correct the error. You're going to write your error and you're going to do your correction. There is no shaded area, so I'm going to tell you to look right here at the experimental setup. What could be the error here?